All right, Stefanie, I'm going to start with you. Thank you for being with us today.
Thank you for having me.
Like Tom, you have an extraordinary story to tell. You were diagnosed with colon cancer at the age of 22. What did your initial, first of all, what was that experience like? And then what was your initial treatment plan?
Yeah, so that experience was a huge shock, of course. I have something called Lynch syndrome, which is a genetic disposition to certain cancers, colon cancer being number one. So I had actually just gone to a primary care and found out that I was severely anemic. And she, knowing the history of Lynch in my family, suggested a colonoscopy. So that experience was me waking up to a gastroenterologist, white in the face like a ghost, just so shocked at what he had seen. And it was a really advanced stage 2, but really advanced colon cancer. That stage 2, so operation, we thought we had gotten everything at that time, and basically no further treatment was recommended. But then 9 months later, it metastasized into my abdominal cavity. And obviously when a cancer metastasizes, it becomes stage 4. It's a completely different story. At that point, another surgery, and then followed by a chemotherapy regimen, FOLFOX, which after 2 months on that, the cancer returned again for a third time. They put me on a more aggressive chemotherapy, FOLFIRI, and at that point, the cancer was just completely out of control and totally inoperable.
And so you must have been around 23 at this time, after you've just been through a pair of surgeries, a pair of what I can only imagine were rather hellish chemotherapy experiences. And then what happened then?
So I'm at the end of the road, and I'm basically told by my doctors, essentially, you know, there's nothing left that we can do for you. Best of luck to you. There's interesting work being done in immunotherapy. That was the extent of the direction that I was given. And I, at this point, am like barely 100 pounds, taking a ridiculous amount of narcotics every single day for my debilitating back pain. I am not living at all. I'm sleeping like 18-hour days, and I'm just not a person. So when you hear from the doctors essentially give up, you kind of are inclined to give up. And I had tried to stay mentally really strong throughout, but it was really tough, obviously. So at this point, actually, my little sister goes home and will not take this "no, it's time to give up" as an answer. So she scours the internet and goes on clinicaltrials.gov and just starts inserting words that we had heard, buzzwords about the sort of genetic components of my cancer. So MSI, microsatellite instability, is obviously a huge biomarker right now. So she found this phenomenal study at Johns Hopkins that saved my life in immunotherapy. Thank you. It still feels very surreal to even talk about.
Were you guys, were you guys very close before then? Like, how's your relationship changed since then?
Jessie and I, she's my other half. I mean, she's my soulmate in this life. We're best friends. We always have been. So it's just very, it just is the perfect narrative because it fits our relationship already before cancer. So she really, we're close as ever, and it's incredible to me till this day. I'll never forget when she came into my room with her laptop open, and I was in a pitch black room, just really demoralized and ready to give up, and I wouldn't get out of bed that day. And she came into my room and she was like, "Steffi, like, I think I found this study. I think I found the perfect trial for you." And I have chills right now thinking about it. And then I sent an email to my first oncologist in Philadelphia, and he just confirmed, you know, indeed, this is a great study, and put me directly in touch with Dr. Luis Diaz at Hopkins, who ended up calling me personally and said, "Get down here as fast as you can. We've been having tremendous success with patients like yourself." And I tell him every time, "You saved my life on the telephone," because that was a lifeline, that hope, you know, just this hope that something could potentially work, and it did.
Dr. Roychowdhury, you work in immunotherapy. That's an understatement to say you run an immunotherapy department. And can you tell me what's the drug in question here? The treatment is called Keytruda. What's special about that one?
So this is the first time that a new therapy has been approved for a marker or a biologic component of a cancer as opposed to the type of cancer. So normally drugs get approved for colon cancer or lung cancer or breast cancer, and what we're beginning to now understand is that there's not one kind of colon cancer. Everyone's cancer is going to be a little different, and understanding why that cancer happens and grows can help us determine what's the best treatment. And so this example, the marker that you mentioned, MSI, that's the approval. So the drug is approved for anyone with a cancer that has the MSI marker. That's a measure of how much repair is evident in the DNA of that cancer, and it makes you more susceptible to an immunotherapy, and thankfully very susceptible in some instances. So these new markers are sort of the pillar of precision medicine. So what kind of lung cancer, what kind of colon cancer, and does that marker tell us about the biology, and does it tell us about sort of the weak point or the sensitive point that we could target with a new therapy, such as immunotherapy or new smart drugs?
Of the percentage of cancers that are out there and known, how many of them have not this marker, but markers like this that we know of and have identified?
That's a really good question. So today, when we do the most advanced genetic testing that we have available, many companies do this, academic cancer centers do this. On average, we think perhaps 4 out of 10 patients are going to have a marker that helps us choose a therapy. But interestingly, the challenge is finding those clinical trials and bringing the drugs to patients. So even though I may find 4 out of 10 patients who have the right marker for a therapy, I'm only able to offer them 1 in 10 that new therapy. So what we have to do is bring markers, tests, and new therapies and clinical trials closer together for those patients so we can offer more patients novel therapies earlier in their lifetime.
And Stefanie, it sounds like you feel like the patients need to sort of self-activate and go find them.
Yeah, well, what's happening right now, which is really exciting, it's the most tremendous time in cancer care and in cancer treatment, but the field is moving so quickly that oncologists simply can't keep up. If I was at one of the best, cancer hospitals in the country, and the physician there, wasn't aware of this trial, it's not a matter of, it's just oncologists can't keep up. The information is coming in every single day, every single week, things are changing. So it's really important for patients to become, as I say, like an expert in their cancers. I don't mean to understand all the science and all the genetics, but really understand what options are available to you, really become empowered to seek out that information simply because what your oncologist is telling you, to their best knowledge, is true. However, they might not know of a small study happening in this or that hospital or the most recent research. So it's critical time right now. The last 5 years have been, I don't even know how to quantify it in terms of the last 75 years in terms of since chemotherapy came into the conversation, but it's been tremendous, and patients need to really seek out and understand all of their options and not just take their doctor's word at face value, because sometimes it's just not the best option.
Yeah. Dr. Rollins, you've got a post at Harvard and a post at the Dana-Farber Institute here in town, and I wonder, that gives you sort of a bird's eye view of this field. Are you seeing lots of stories like Stefanie's? Is this something that's happening more commonly?
Well, we're seeing more and more stories like Stefanie's, and what that reflects is, just as Stefanie said, this is the single most exciting, rapidly moving era in the history of cancer research or cancer treatment or cancer care that any of us have ever seen. I first started working in cancer research in the mid-1970s, and the change between then and now is truly unbelievable. And the driver of that change is basic science research. Yeah. The biomarkers that we have been using up to now, you mentioned biomarkers, things like mutations that drive cancers, those discoveries were made by curiosity-driven, mechanistically based research. The drug Keytruda is directed against a target, a protein that was discovered at Dana-Farber in the mid-90s by somebody working on the fundamentals of the immune system. He wasn't really working on cancer. He wanted to figure out why, if you get an infection and your immune system goes after this infection, why does it quiet down again? You realize that it has to quiet down again, otherwise you're in big trouble in terms of inflammation and autoimmune disease. And he discovered these proteins that do this. Totally basic science stuff, but within 10 years it became clear that these were the proteins that suppress our own immune system's ability to attack cancers. And that's why Keytruda and drugs from BMS as well as Merck and several other companies are now so effective. It was based on basic science.
Is there a range when you see patients that are looking for clinical trials or who are entering clinical trials, is there a range of, I mean, it sounds like Stefanie was quite an enthusiastic patient. Are some people more hesitant? Do they worry? Is it a really stressful process for people to enter these trials?
I don't think it's stressful to enter the trials. I think it's stressful to get a diagnosis of cancer. I think holding out a trial is a way to actually think about being active, being an active participant in your disease. I think we have to be a little careful here. There is something called the therapeutic fallacy, which is that a trial is really treatment. You know, if it were treatment, it wouldn't be a trial. We wouldn't be doing the experiment. But in cancer, you're in this funny sort of mid-ground where an experimental therapy can be thought of sort of as a therapy as well. But we have to be careful not to oversell the idea of an experimental therapy until we really know that it works.
And how are these trials usually funded?
Well, I would say, and probably it's true in your institution as well, the majority of them are funded by the drug company who wants to get their drug approved, so-called sponsor-initiated trials. But increasingly, as we get to thinking about cancer as a disease of genes, which it is, it's not a disease of organs, it's a disease of genes, and as we think about drugs that are indicated for pathways, like the MSI pathway, individual investigators are coming up with the ideas of these trials. And here, that's a struggle. You know, we try to get funding from the National Cancer Institute, we try to get funding from the American Cancer Society, we try to get funding from philanthropy. That's a really tough row to hoe, but we try.
Is any of this research, and I don't just mean the trials, I mean the kind of basic research that you were talking about that led to Keytruda in the fullness of time, is that in peril due to some of the cuts that we're seeing in proposed budgets by the Trump administration?
How much time do you have?
Well, yeah.
I'm sorry.
No.
12 minutes.
I'm not going to take 12 minutes. I just want to make the point. I previously said this is the most exciting time in the history of cancer research. This is the most imperiled time in the history of support for cancer research. From 1998 to 2003, the NIH budget doubled. And the NIH budget is the budget that allows this basic research to take place. Most of the other sources of revenue are really more directed and more applied. But that's what supports basic research. Since fiscal 2004, the NIH budget has dropped, either in nominal terms or in constant dollars, every year. And now it is actually 25% lower in real terms than it was in 2004. The proposed budget, which is a further almost $6 billion cut, brings us to levels below 2002. The problem is with that proposed budget, because of commitments, we usually give grants in multiple years, 4-year, 5-year grants. Those are committed dollars. If this budget gets passed, no new grants for the coming year. None.
And Stefanie, you've been part of this fight a bit.
Yeah, absolutely. I actually had the privilege of attending a House and Government Reform Committee hearing on federally funded cancer research. I went with Dr. Elizabeth Jaffe from Hopkins, and basically the committee just wanted to hear, you know, the argument for not cutting the budget and you know, hopefully increasing the budget as was expected with the moonshot, with the $2 billion yearly increase. And it was really impactful for me to understand, you know, how important it is for citizens, for patient advocates, and for all of us to be talking to Congress and to tell them the importance of this. It was really moving. Dr. Jaffe and I, after the hearing, went around to representatives and just spoke to them, and it was really, I think, impactful for her to say, hey, this is why we, you know, this is all the research that I'm doing, and this is the clinical benefit, and I'm here because of decades of basic research and of basic science, and I will never forget that. It really has. Everyone's like, oh, why are we only now paying attention to the immune system and immunotherapy? No, it's been 30+ years of a lot of basic science and investigative work that is mostly funded by the NIH. So to even conceive of cutting it almost by $6 billion is unimaginable. It should, and thankfully, I think that it's generally a bipartisan agreement there, but hopefully it gets increased by much more than $2 billion.
So one of the risks of losing funding is not just the science that we're funding, but it's the young innovators.
Right.
So the persons that are going to make the biggest impact are young scientists, and their average age for the highest level of innovation possible, you can look at this and study this, is around 25 years of age. Early in science, crazy ideas that neither of us would think of, but that's why we do science. We're trying to solve problems we don't know the answer to. And so that's what's at risk. When they see how difficult it is to secure funding, when grants are submitted, 1 in 10 will be funded, and it can take up to 24 months to get the funding back, actually. And so that process is very risky for young investigators, and they are walking away. They see what's happening. They see what's happening in Washington. So those dollars are part of human capital, and they're part of research capital. We need both of those capitals to make science happen.
Yeah, we're at risk of having a completely lost generation of
Right.
cancer scientists and general scientists.
Are there places around the world, like for instance, new President Macron in France has just invited climate researchers with quite healthy grants from the U.S. to come and do their work there. Is there a similar kind of thing in the international community where can people who are leaving the departments, can they go elsewhere and do this work?
They can. I think that even in our straitened circumstances, American policy has been more generous to research than any other policy around the world. There are lots of dollars being spent in China. There are lots of dollars being spent in Europe, but as a percentage of what's available, not as high as here. And why would we want to cede our leadership in the first place? I think I have colleagues who've gone to the UK. I have some colleagues who've gone to China, and it's not that they've regretted it, but they do it in a rueful sort of way. I mean, they would have liked to have continued working in this environment, which is pretty special.
Now, both of you are involved in not the same one, but different data sharing programs, and we've heard a lot of hype about that of late, justified hype maybe, about how pooling data together across different institutions can lead to some really low-hanging fruit for researchers. Have you seen that already?
So one of the initiatives that the James Cancer Hospital and the Moffitt Cancer Center have started is something called the Oncology Research Information Exchange Network. So the acronym is ORION, so we're going to reach for the sky here. And what the idea of this ORION Alliance is, it's a partnership between patients, hospitals, and industry. So those are the three partners that need to be involved in science and drug development for cancer. And so when a patient signs up for this study at one of more than 15 different hospital systems, which is growing as we speak, they agree to give their cancer tissue to be profiled in any way possible, to have their clinical information shared for the alliance, and then finally they want a phone call, and they agree to that. So here's my tumor, here's my information, call me if you have a trial for me. So this would be something that you might have signed up for and said, hey, if you've got something that fits me. And so this is a two-way, it's a three-way partnership, but a two-way goal, right? So patients are looking for the right drug for them, and then pharmaceutical companies are looking for the right patient for their drug. And so this concept is going to allow us to bring new therapies, for example, for a marker that may be only part of 4% of cancer across the board. This network could help us find those patients, offer them the drug in a new clinical trial, and we can bring the drug to them. And the network helps us find those patients. And that alliance kind of helps us solve one of the funding problems we have by kind of partnering with both patients, hospitals, and pharma. We can do that, but it doesn't solve the problem, but it helps us get there.
So Orion is a great example of something that aggregates data and is looking towards clinical trials. We're involved in something that's a little bit earlier. This is something called Project Genie. If I could remember what the acronym stood for, you know, I could have really scored, but I don't remember. It's genetic something. But this is a program sponsored by the American Association for Cancer Research. And in its first instance, it involved 8 of the largest cancer centers in the country and around the world. And we have all pledged to take all of the DNA sequencing data that we have gathered on all of our patients that we all sequence clinically, and we pledge to combine all those data in a single database. And after about 6 months to a year of exclusive use among the consortium members, we release it completely to the public, along with a really easy user interface for anybody who wants to delve into those data. And the reason they're important is what you started to bring up. We know that there are common mutations, and then there's a long tail in the curve. And that long tail contains mutations that are really important. 2% of lung cancers have a certain kind of mutation that there's an incredibly effective drug that treats those patients. But you have to know about that. So in the last public data release, we did our first public data release in January, 17,000 profiles, and importantly, all linked to clinical information. And so we can start to discover new abnormalities that are associated with different diseases and then feed that information to something like Orion, which can then start to match patients with clinical trials. We are in an age where people can't complain anymore about how we don't talk to each other. I've spent, I don't know, 20% of my time and effort on the phone or on airplanes, going to other places, trading our data, opening up our data, making sure that everybody can use it in a safe and a safe way that honors patient privacy, but in a way that shares it worldwide.
I want to allow a little time for our audience, but before I do that, Stefanie, how has this experience been after the trial, and you've become this sort of advocate for patients, and your experience on Capitol Hill sounds great. Not many people have a fun time up there, but out in the world, talking to patients, are you finding people? How are people responding to your story?
Yeah, it's been pretty phenomenal. I mean, I still pinch myself every single day that I'm even here. It feels surreal to even talk about it. It feels like I'm talking about somebody else's story. I mean, if I were to have gotten as sick as I did, even a few months prior, I would not be here today. So I'm incredibly grateful and wanting to use my story and my voice to help empower other patients to know that there are other options, that just don't be disempowered by your doctors. Seek out different options and different clinical trials. Understand that you are a partner of science, that they need us a lot. Only 3 to 5% of patients participate in clinical trials because there's just a big misconception in the public that clinical trials, and I was totally guilty of this, thinking that clinical trials are a last-ditch option or a last resort. And actually, we're in a time where clinical trials could be the most precise treatment and the most innovative for your specific cancer. No two cancers are the same. So I've found that it's been tremendous, and the feedback that I get is great. I think it feels nice for people to feel like understood in terms of having an advocate who all I preach all the time is patient empowerment, because I would not be here today if I didn't empower myself with knowledge, if I didn't, of course, have a family and a sister who fought so much for me. So it's been a wonderful transition, a very meaningful life. So I am now transitioning from a digital marketing career into a full-time advocacy career, and
cool.
I'm thrilled. Yeah.
Very cool. Yeah, I want to open it up for questions. Oh, no questions. Okay. Oh, there we go. Sorry.
How many years are you cancer-free?
Thank you for asking. I am NED, which is no evidence of disease, since February 2016. So a little over a year and a half almost. I went off study in August of 2016. So I was on the immunotherapy for a full 2 years. Within 3 months of my first infusion, my tumor shrank by 65%. Within 3 days, my pain started to ebb completely. It was a surreal response. Essentially what that means is like the lymphocytes, everything was ready to go. It just needed to take the brakes off of the immune system. So on the receiving end, it feels like a miracle. I understand that decades of science went into it, but it felt like an absolute miracle to sit at the dinner table within a month of my first infusion and cry because I was hungry. I hadn't experienced that in a long time. So yes, I'm cancer-free for over a year. Thank you.
Yeah. That's a great note to go out on. Stefanie, Dr. Rollins, Dr. Roychowdhury, thank you for joining us today. It's wonderful.
Thank you so much.
Thank you.